There are various chromatographic techniques. They are paper chromatography, thin layer chromatography, gas chromatography, high performance liquid chromatography, or high pressure liquid chromatography, gel permeation chromatography, ion exchange chromatography, etc. There are a lot of chromatographic techniques. So nowadays UPLC is also there. There are a lot of chromatographic techniques in which we are going to discuss about ion exchange chromatography. So basically the introduction. So ion exchange chromatography is a process by the mixture of similar charged ions that can be separated by using ion exchange resin. So these resin are based on anion exchanger or cation exchanger. These are the resins. So I will explain that in the next steps. Ion exchange resin exchanges ion according to their relative affinities. Means here the separation takes place on the basis of charges, their charges, positively charged or negatively charged. This takes in very important role in ion exchange chromatography. It is there is a reversible exchange, means it can be reversed, and most similar charged ions like cations or anions can be separated by this technique. So it is used for almost any kind of charged molecules, for example large proteins, small nucleotides, amino acids, enzymes, etc. Can be used, or hormones can be we can use to separate. Ion exchange chromatography preserve unallied molecule on the column based on ionic interaction. As I said, positive and negative charges takes place a very important role. This is the one of the role. Here in every chromatography, the important things are mobile phase and stationary phase. Here, as I said, the stationary phase is resin, and the resin is not only that it is having the only positive sign. The stationary phase may be of positive charge or may be of negative charge. The mobile phase is buffer solution in which the pH and salt concentration changes according to the elute or according to the sample solutions or according to the molecule which need to be separated. So ion exchange chromatography is based on charge. So first of all, let us study about cation exchanges. So here in the cation exchanges possesses negatively charged groups. Means as we know that cation means positive charge. Okay, so positive charge will always attract the negative charged functional groups or molecules. This is cation exchanger. So next one is anion exchanger. So it means it exchanges anion. So here I can tell you by using this example. So here you can observe the first one, which is in the blue ball, is anion exchanger. Means it exchange anion. Anion means negative charge. It exchange or it attracts negative charge from the sample by using the anion exchanger. Anion means anion exchanger consisting of positive charged particles, means cation particles, because it need to attract the negatively charged particles. That's why it is called as anion exchanger. In cation exchanger, it attracts positive charges, but the resin bed consisting of negatively charged particles. So this is how the ion exchange chromatography works. So let's move to the properties. So they are complex and polymeric in nature. These are the properties, and it is insoluble in water and soluble insoluble in organic solvents also, and they contains active ions which can be used to reverse the phase. So next to the principle, as I said, I already moved to the principle. Anyway, by using this diagram, I will explain you about the principle. So the all ion exchange chromatography takes place in a column like this. This is the column which we used in the ion exchange chromatography. So this is the blue colored particles are there now. They are called as resins or stationary phase here. So this stationary phase attracts, here you can see that it is a negatively charged stationary phase, which means it is a cation exchanger, means it attracts the cation, so that it is made up of negatively charged particles so that it can attract the positively charged cations. So here you can see that the sample, it consisting of a sample along with buffer solution is adding to the column. Here in the column, here the resin is cation exchanger. So all the cations will be stored here, and the remaining anions will be flowed through the elute. The first step is the negatively charged anions flowed through the elute, and then only the positively charged, whatever the sample consisting of positively charged cations, will be stored here only. Then the buffer solution and the concentration of salt changes here. So then only it will get detached from the resin beds or stationary phase. Then it will get as the elute. The first one, when the anions come eluted first, then it will be separated in the separate chamber or separate beaker. Then only the concentration of the sample is changed, the buffer is changed, and then the the positive ions or the cations will be eluted, then it is separated, it is collected in the separate beaker. Okay, that's how we can separate the positive ions or the positive molecules and negative molecules. This is how the ion exchange or ion exchange chromatography work.